Hi, today we are going to be talking about metabolomics. Metabolomics is an actually very complicated topic. I'm going to try and do my best to make it as easy as So what is metabolomics? Metabolomics is the study of the chemicals process involving metabolites, which are small molecules that are substrates. They're mostly going to be like your lipids and amino acids, things like that. And at the endpoint of the physiological and pathological process, depicting both the genetic predisposition and the environmental influences like nutrition and exercise and medication. Metabolomics literally is your entire body's processing. Think about your metabolism. So this is an omics cascade. This is more of a general picture of all the different types of omics. So genomics, proteomics, transomics, and metabolomics. So it starts out with what can happen. We start in the genomics, which is your DNA and your RNA, which goes through transcription. What appears to be happening is we're trying to make proteins. That's the ultimate goal. And if we have like a cancer, then it folds over weird, and then you have an entire mess. What makes it happen is that the proteins are folding on top of each other in the correct way, and we are creating a small molecule for metabolomics. So it's part of your metabolism. And all of this is basically the central dogma of biology, DNA, RNA, protein. That's the end goal. Metabolomics is, again, the study of substrates and products. So genomics is genes, transomics is mRNA, proteomics is proteins, and metabolomics is metabolism. The small molecules, they'll range from 50 to 1,500 Daltons, which is teeny tiny. So again, think about your sugars, lipids, fatty acids, things like that, and look at how these are all structured. They have similar structures, and they have similar OH or HO groups and OH groups. That makes them part of your metabolism. It's everything that goes in from the second you start eating all the way through your stomach and all the way through your entire intestines. Also, metabolomics has a really big contribution to your muscles and how they function. So anemia is a complete set of metabolites within a cell, tissue, or biological sample. Those are everything. You can just take a part of your cells and see that. These reactomes are freely available, curated database of the pathways and reactions. This is so good for us to understand. We can add a medicine and make it better. Some people are susceptible to certain foods. It's all part of the metabolism. Again, the series of chemical reactions catalyzed by enzymes where the product of one reaction becomes the substrate for the next reaction is divided into anabolic and catabolic, which is part of your metabolism, how we metabolize. So why is it important? It is the ideal tool for pharmaceutical and preventive healthcare and agriculture. Again, if we know what we're looking at, we can try and help it. For those who have issues with certain medicine, if you can't take ibuprofen or if you can't take Tylenol, we can look at how it's metabolized in the body in order to see where it's going and how it is affecting. In agriculture, we can create new pesticides. This helps get rid of a bunch of different kinds of issues. So people are not happy with certain steroids. So people are not happy with the pesticides because of the toxicity in it. Also, we have indirect targets whenever we use pesticides. So we're not just hurting, like for an example, the locust. We end up hurting everything else, like the locust's primary predator or things like that, and we're making we're making a lot of things more toxic than we need to. So with metabolomics, we can make it easier for us to not make our environment so toxic. Biomarker discovery is distinguishing two groups of samples, typically a disease and a control group. For example, a metabolite presents in disease samples such as urine, saliva, bile, semifluids, and things that contain high form of metabolites. Think about it. Whenever you metabolize, where would you find most of the product after you've eaten? In waste. So again, we're looking at what we have. We have an untargeted approach and we have a targeted approach. The untargeted approach is the method that is global. It is a range of biological samples within a basis. And we have targeted approach. This method is where you want to use a measured set. So whereas it's more general or very specific. How we can do that, you can set up an experiment, think about regular plain old experiment designs, and this is how you'd go about it. All right, some background. Sir Archibald Arwin Garrod was one of the first ones to start really looking into it. He was a postgraduate. He did publications on chemical pathology and one gene and one enzyme hypothesis. He was a very big contributor to this. So here is an experiment. They were taking newborns. They were screening them for metabolic disorders. What they were doing is they were replacing expensive monoparamic assays. This allowed for them to simulate detection of 40 to 60 different metabolites, so different amino acids and lipids and things like that. The diagnosis required a 20 to 30 monogenic disease. In total, instances was 1 to 2,000. However, it became easy to find, and we were able to discover that we could do that. So 1.3 million newborns were screened, and similar labs worldwide continue to do this because we can find out their metabolism now. We can look at how they metabolize as a newborn infant in order to help them as they grow. So what did we learn? Quantitative mass spectrometry, that's a hard one, is able to meet the most stringent quality of the criteria. So mass spectrometry is going to be this big giant graph. It shows you all kinds of different things about all the different little molecules and enzymes and stuff. It's a little bit overwhelming at first because, again, this is a very hard topic to understand. So here's a case study. Or current studies. Rapid metabolic shifts occurring during the transition between hunger and not having hunger. So like our metabolism, people talk about how their metabolism is slow or their metabolism is really fast or they only eat like once a day versus 3 times a day. That's going to depend on that's how we study this stuff. We take somebody that's like, oh, I only eat once a day and I'm fine. Or somebody who has to eat constantly or like grazing. That's how we start these studies. But this is all the different things that it takes, or different ways we can look, all the different ways of technology of making things. You make a sample preparation, you have to analyze it, then you use bioinformatics. That's what most of this class is going to be for. And this is how you would try to navigate your workflow if you wanted to. This is an example of a diabetic and non-diabetic neuropathy based on metabolomics. Yes, you may not understand what it looks like right now, but all of these numbers can show us the difference between the metabolomics of a person that is diabetic or that is not diabetic. And the neuropathy, that's a very big deal for somebody who's diabetic. That can cause so many issues if they're not careful. They have to take very good care of themselves for that. Again, this is another example of how we can use it for stage 3 and stage 5 kidney diseases. Look at those numbers. Look at how they range. If we know exactly what we're looking for and what protein or what lipid that we are looking for, we are so much better off to be able to look at this giant number and see how we can see the differences in stage 3. Again, here's another example of oxidation methane. Is highly indicated for oxidative stress. So much stress when you're trying to breathe. Again, here's another example. All right, so this is what fatty acid oxidation looks like. That is where we break down those fatty acids that we eat, those lipids and things like that. See, we go down and then up. We battle it. Here's the web. Exploring metabolic shells around the metabolites, there are so many things that go into one. So that one metabolite has so many different things that may be a receptor or can break it down or act as a catalyst, or it can be targeted. There are so many things, and we don't know everything about them because there's so much to study. It's very hard to pinpoint just one. When we think about our programming and things like that and the databases, it's very hard, again, to pinpoint one issue because there's so many things that can be a product. Like I can break this down and it turns into an acid, or you can break something down and it turns into something else, or we have multiple products from one metabolite being broken. So there's a standardizing and a normalizing, and the standardizing is more data formats and what can we do with the records, and normalizing is just more quantitative overcoming. And there's so many biostatistics. We can run a lot of statistics, but again, as much as statistics as we run, we still are not always able to pinpoint exactly what we need. So if we keep a giant database of all the statistics, maybe we can figure it out. Maybe we can create algorithms. Maybe we're able to create really good graphs. We will figure. All right, so let's sum it up. So what did we learn today? Metabolomics depicts the function endpoint of genetic and environment. So your products, what happens when you eat something? You get energy, you make ATP, you have sugars. The targeted metabolomics of data is that analytically reproducible and allow immediate biochemical interpretation. So that being said is that once it makes that product, it can be interpreted instantly. We know exactly what it is. Proof of concept has been achieved in routine diagnostics of inborn errors of metabolomics. So that saying is that we know that we can find the issues in metabolomics from a young age. We can screen newborns. Many metabolic biomarkers are valid across species and enable translational research. That means that we can look at like how a pig metabolism works and almost compare it to our metabolism. We can do all kinds of things looking at it. Oh, plants metabolize much differently than we do, but we can still look at it and have some kind of concept and idea of what our similar metabolites are. Comprehensive targeted metabolomics bridges the gap to proof of open profiling approaches. So we can profile each of these, put them in their own zone. So here we go, looking at what we can do and how we get the biomarkers, and then we validate them. Always validate everything. Thank you. I hope that you learned something today.